On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Dynamic Biosensors, I'd like to welcome you to Target Selectivity of Multispecific Antibodies, Practical Analysis of Binding Kinetics and Avidity. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I would like to introduce our moderator for today's webinar. He is Dr. Anthony Ferrante, Sales Manager with Dynamic Biosensors. Welcome, Dr. Ferrante.
Thank you, Elizabeth, and good morning, everyone. As Elizabeth mentioned, my name is Anthony Ferrante, and I'm the U.S. Technical Sales Manager for Dynamic Biosensors. I'm based in our Woburn, Massachusetts office, where we also have a laboratory to run customer samples and to welcome customers for on-site demos. I would like to welcome you all to today's webinar, which will be presented by my colleagues, Joanna Deek, who heads U.S. Business Development, and Application Specialist Stephanie Mack. Today's talk will introduce our unique biosensor platform that enables characterization of a variety of bivalent constructs, all using the same workflow. We will use our large body of data generated using bivalent and bispecific antibody constructs to illustrate the flexibility in surface design that enables us to work with analytes having different features. And now to tell the story are Joanna and Stephanie.
And with that wonderful opener from Anthony, I look forward to demonstrating our workflows for assaying bispecific binders. The most prominent example is traditional IgGs, including bispecific and multivalent formats, and more recently, constructs that link two binding modules such as DARPins. In addition to large macromolecules, we can also work with smaller molecules such as PROTACs and that have been developed for proximity-induced drug development. The common feature for all of these analyte binders is the presence of two or more binding sites in close proximity. To interrogate the binding and interaction properties of bispecific binders with confidence, various practical aspects relating to the assay design and setup are needed. More specifically, to control, for instance, target ratio and density in the instance that you'd like to use more than one target when working with a bispecific analyte, and also to detect cooperative binding, in this case to see how the individual binding arms behave in presence of their single targets, but also how the cooperative effects of combining both of those binding modules into one impacts the overall construct. Of course, having a wide range or being able to resolve a wide range of kinetic rates only means that you can extend an individual assay to look and explore various different targets that you have in your workflows. Flow rates to prevent mass transport limitations and rebinding are of course considerations when working with much weaker binders. So let's start with some state-of-the-art technologies that are typically used for the analysis of bispecific binders. I would like to start with solution-based technologies, and that are used, of course, very frequently within the community, and as a lot of our attendees had indicated, they have quite a bit of experience as well with ITC. Now, while this, of course, presents a lot of advantages and has done much in order to advance our understanding of bispecific binders, it is rather challenging to assay, for instance, cooperative binding in this case, specifically because you cannot put or detect the cooperative effect of two different targets together at the same time. This is, of course, an equilibrium measurement, which means that you cannot assay on rates and off rates, and therefore considerations such as mass transport limitation and rebinding don't come into play. Nevertheless, the inability to have both targets present when working with the bispecific analyte are alleviated when working with surface-based technologies. In this case, to use, for instance, SPR and BLI as an example, you can immobilize or functionalize two different targets on the surface, and by that you're able to assay the binding of an individual analyte to the targets simultaneously and observe how the presence of both of these targets actually impacts the binding affinity in this cooperative and avidity binding mode. There, of course, is also the ability in this case to resolve a wide range of kinetic rates. There remains a slight consideration with mass transport limitations because of the limit with flow rates when working with these technologies. And of course, here is where we would like to come in, working with the switch sense technology. In this case, again, it's a surface-based immobilization technology And the readout is fluorescence. In this case, fluorescence is very forgiving, which means that we can work with a very broad range of flow rates, and that enables us to address with confidence, again, a wide range of kinetic rates without concern that we will have any mass transport limitations or the potential of rebinding in the dissociation profile of our targets. Now, unique to our SwitchSense biosensor is the ability to very accurately engineer or tailor the surface that we have in order to generate surfaces not only of one target and two targets, but also targets in variable different ratios and densities. So understand that, please, as abundance, relative abundance of two different targets on the same surface. Expanding on the surface immobilization features and using the Helix user interface, you can take advantage of the patented microfluidic system to resolve rates, as I mentioned, without rebinding events and mass transport limitations. You can measure using very high flow rates to resolve fast kinetics, but also for very long time windows to observe surface recovery for slow kinetics. These features, in combination with the specialized software for automated data analysis, allows for the real-time measurement of kinetic rates of bispecific binders. And to illustrate what a typical readout looks like, I would like to walk you through a standard bispecific antibody binder SwitchSense assay that measures the affinity binding of single-arm kinetics, as well as the bivalent and cooperative or avidity binding in the presence of both targets. In the profile or in the global concentration series that you see on top, these are our sensorgrams for the binding of a bispecific analyte to one of its targets. In this case, again, as a function of increasing concentration, the signal amplitude corresponding to the binding rate, as well as the binding amplitude of the antibody itself. After a rapid association, you see over here, this is probably reflected or easily seen with the concentrations of the analyte, which are in the micromolar range. There is a very rapid dissociation of a bispecific analyte from the surface. Now, in presence of both targets, we see a rather different behavior. In this case, of course, we associate it onto the surface, again, using the same concentrations in the global concentration series, but need to dissociate for a slightly longer period of time in order to observe full surface recovery. Now, the biggest distinction between these two plots, of course, is at the earlier times for the dissociation, and here what this typically corresponds to is the avidity enhancement, or the enhancement in the binding of the bivalent antibody in presence of both of its targets. So you basically hold on to that analyte for a longer time on the surface because it's more strongly engaged. Now, our software, the Helios software that I had mentioned earlier on, allows you not only, of course, to image, to generate these curves that you see before you, but also to run the analysis of your data. In the first case, it was a mono fit, so a single phasic fit, in which we could generate both the K-on rate and the K-off rates for the binding to the single target, and then running the analysis, in this case, using a biphasic fit in order to show not only, of course, the contribution of the single engagement of the target on the surface, but also more notably the avidity engagement. And as you see that over here, there is a significant shortening, meaning a reduction in the off rate. So the antibody remains on the surface for a lot longer. So now that we have explored the readout very briefly, and of course we will get to that in further detail with the assistance of my colleague Stephanie, I would like to describe the workflow behind the empirical realization of these biosurfaces that actually enables us to run these various assays by controlling which targets are immobilized on the surface and which type of assay orientation we would like to use. So to start with the standard biochip surface that we have. You see that represented right before you in the center of the screen. The way that we functionalize our surface or create the functional surfaces is using DNA-encoded addressing. This means that each of our biochips comes with covalently attached non-modified DNA. So it means they don't carry any fluorophores or they don't carry any ligand. What we do is that we create a premix or a ligand mix which carries the fluorophore. This is the reporter fluorophore that we use in order to monitor the binding kinetics, and that is pre-hybridized with DNA that is attached either covalently or using capture methods to the antigen of choice. In this case, we're going to refer to it as ligand A. This solution is pre-hybridized, flowed over the surface. DNA hybridization is highly efficient. As a result, what we do is that we immobilize this ligand onto the surface. So from a non-functionalized chip, we can create a functional one using minimal sample and a very quick assay workflow. Now, one important parameter to vary when working with antibodies is to be able to modulate the monovalent versus the bivalent engagement of your target. And in this case, what would be of interest is to have some control over the target surface density. Here, this is to be understood as the average separation between the surface immobilized ligands. This can be rather easily realized on our biochip surface by adding a non-modified, what we refer to as a dark adapter strand. So this is, again, a DNA sequence which is complementary to the anchor strand that we have on the surface, which is flown over. So what that does is that it hybridizes onto the surface. It creates what we refer to as dark duplexes. These dark duplexes do not contribute to the fluorescent signal that we use in order to monitor the binding kinetics. All it does is serve to separate between the different DNA sequences that we have on the surface. So effectively, what we understand density to mean the relative separation of these antigens. Now, of note, the density or the separation that you have on the surface is a direct function of the ratio of these two solutions that you mix in the vial beforehand. So if you have a 50-50 ratio of these, you can understand the surface to be diluted 1:2 or 1:1, excuse me, on the surface, and then you effectively increase the separation between your two antigens. And here we wanted to demonstrate how, or the linearity between the relation between the ligand concentration in solution as well as the fluorescent signal that we observed on the surface for each of these solutions. And as I mentioned, as you continue to dilute your ligand solution, in this case, fluorescent carrying ligand solution with the dark adapter strand, as you continue to dilute that, you're also reducing the fluorescent signal that you see on the surface. So there's a 1-to-1 correlation between those two. Now, DNA itself can either be, since we're running all of these assay setups using pre-hybridized mixes instead of using a dark adapter strand, for instance, and more specifically, as would be relevant for the constructs that we are prescribing today, it would be to have two different ligands on the surface, so two different kinds. In this case, they would be pre-mixed with different in different solutions. In this case, ligand A will be hybridized, for instance, with an adapter strand that carries a green fluorophore, and ligand B will be pre-hybridized with an adapter strand that carries a red fluorophore. And I'll explain shortly the relevance of both of these fluorophores. But again, in the same way that we had mixed them together in order to generate different densities using just a standard adapter strand and one ligand, here you could mix two different ligands together in order to create bifunctional surfaces. So surfaces that have two different ligands. So in this case, you can assay both affinity and avidity binding using the same assay orientation for your bivalent analytes. And again here, the ratio with which the two ligand solutions are mixed together will correspond directly to the ratio that you see on the surface. Now, to jump to these fluorophores that we had mentioned earlier, and which is one of the strengths of our Helix biosensor, we have two different LEDs, which means that we can illuminate both of these fluorophores simultaneously, and we have 4 single photon counters that are able to generate the readout from each of these channels. The advantage here, of course, being is that if you have two different fluorophores immobilized with two different ligands on the surface, you are able to observe simultaneously two independent binding events. So we can actually look at the binding of a bispecific analyte to one of its ligands or both of its ligands on the surface at the same time. Extending this even further, we can also play around with the overall density that we have of ligands on the surface as well. So not only can we mix two different types and change the density in separate modules, we can also combine both of these two functionalities into one to create various different surfaces that will be relevant for the cell mimicking application that Stephanie and I will talk to you about later. And Beyond the empirical setup, we would also like to show you the mechanisms that dictate the dissociation profile and how the readout is analyzed to reflect the surface behavior of these analytes.
Yes, we will continue with a short excursion into the theory behind our analysis. Here I'd like to walk you once through the underlying models that we use to extract meaningful parameters from the fluorescence curves. This is a process that is normally done by our Helios analysis software in a fully automated way. This is just to show you that we are not talking about magic here. This is a process that's quite easy to understand. A bivalent analyte can bind to a target functionalized surface in two different modes, either with one or with both binding arms attached to the targets. If only one binding arm is attached, the resulting binding is weaker and the dissociation is faster than in the other case. We call this the affinity mode. In the avidity mode, with both arms attached, the binding is usually stronger and the dissociation slower. However, both binding modes can be described by a mono-exponential model, from which the strength of the interaction can be derived. But what exactly defines a mono-exponential curve, and how is this information extracted by our software? A mono-exponential curve is defined by one simple and important characteristic. Exponential curve is defined by one simple and the half-time of the dissociation is always constant. In this example here, it takes about 10 time units to go from 100% binding amplitude to 50%, then another 10 time units to go to 25%, and so on. This constant half-time is the most important feature that our analysis is based on. Usually, in order to characterize the strength of an interaction, the dissociation rate K-off is used, which is inversely proportional to the half-time. The stronger the interaction is, the longer the half-time, and the smaller the dissociation rate. As already mentioned, both the stronger binding mode with both arms and the weaker binding mode with only one arm attached can be described by such a mono-exponential curve. The only difference is the binding strength, indicated here by the very different half-times. On the Biochip surface, however, we rarely can observe the pure affinity or avidity binding modes, but rather a mixture of both. This is dependent on many different parameters, such as the binding strength or the surface density. As a result, we do not see a mono-exponential measurement signal, but a bi-exponential one. Mathematically spoken, this is obtained by simple addition of the two underlying mono-exponential curves. Here you see in pink the fast dissociation curve from before, and in orange the slow one. Please observe how the half-time of the dissociation is not constant anymore. If you see such a pattern in your measurement data, it is always a strong hint on biphasic behavior. In this first example that I have shown you here, the contribution of both binding modes was the same, but this is not always true. If, for example, the surface density changes, either affinity or avidity can become more prominent. Lower densities favor affinity binding, whereas higher densities favor avidity. Here you see a real-world example of a monospecific IgG molecule binding to targets at different densities. We obtained 3 different measurement curves, that is 3 different densities from low to high, and those curves were fit with a biphasic model. From all 3 curves, we could extract the same affinity and avidity off rates, as it is always the same interaction that we watch. Only the contribution of both binding modes changes. At a low surface density, there is mostly affinity binding, because the distance of the target molecules is too big to be bridged by an IgG molecule. Once the density is increased, the avidity binding mode takes over, and the majority of IgG molecules are bound with both arms. The ideal surface density to obtain good biphasic curves, from which both off rates can be determined with confidence, depends on the target size, among other things. Here you see example curves of antibodies binding to targets of different sizes. The smaller the target, the more the affinity binding prevails, here in darker gray. as the distance between the target molecules increase. For very large targets, such as HER2 with about 70 kilodalton, there is nearly exclusively avidity binding, already at a medium surface density. Here we didn't even have to measure at high surface density because we already got nearly 100% avidity already at medium densities. Now we want to use what we learned from the analysis of a monospecific analyte, the single target functionalized surface, and apply it to a bispecific analyte binding to surfaces functionalized with either one of the single targets or to a surface with both targets immobilized at a 1-to-1 ratio. First, we start with the easier setups. Here are setups where the single targets are functionalized on the surface of the adapted chip. Here you see the fluorescence curves recorded by the green detection channel of the helix instrument, which follows binding to target A. The target A is located next to the green fluorophore. That's why you see what happens in close proximity in the green channel. The curves resulting from this experimental setup show a simple 1-to-1 binding kinetics. In the first few minutes, you see the association of the different concentrations, followed by a fast dissociation. The dissociation curves could be accurately fit with a mono-exponential model from which the Helios software then extracts the dissociation rate. In this example, you can see quite a fast dissociation. In less than 10 minutes, the analyte is predominantly dissociated from the surface, effectively exhibiting a half-life of about 1 minute. In a second experiment, target B only is immobilized on the chip. Our bispecific antibody can now bind with its other arm. In this setup, the ligand strand carries target B, located in close proximity to a red fluorophore. The fluorescent signal is captured by the second detection channel of the helix instrument. Again, you see a monophasic dissociation fit with a 1-to-1 model. However, this time the dissociation is much slower. Even after 60 minutes of dissociation, there is a big part of the analyte still present on the surface. This corresponds to a half-life of about 1 hour in this case. The results from these two experiments demonstrate that the affinity of binding arm B to its target is much stronger than that of binding arm A. The important question we are left with now is, how do those two binding arms interact in presence of both of the targets at the same time? In order to answer this question, we would immobilize both targets on the surface at the same time. Now the bispecific antibody can bind to either target A or to target B or to both targets at the same time. All of those binding modes contribute to the overall dissociation profile, but don't worry, I will walk you slowly through the analysis of the separate fluorescent signals for the green and the red channel. The green detection channel that allows you to observe the binding events to target A shows a clearly biphasic dissociation. More specifically, in the first few minutes of the curve, the fast dissociation dominates of analyte molecules bound by only one arm to target A. Slow dissociation then becomes more important at the later time points. Those are the antibody molecules that have bound to both targets at the same time. The red detection channel, here on the right, allows you to observe what happens to target B at the same time. The curves show again a biphasic pattern, not as clear as in the green channel because the two underlying rates, the fast one and the slow one, are more similar in this case. Please observe here that the binding by both arms at the same time has become much stronger. Here we need to dissociate for more than 5 hours to resolve the binding kinetics. Those long dissociations are no problems with the helix instrument, and they are really needed to analyze bispecific antibodies. In the rate overview, you see clearly that the fast dissociation from both biphasic curves corresponds to that from the single targets, both for target A and for target B here in green. In this instance, additionally, the avidity off rate of the green and the red curves is the same, as it represents the same binding event. In conclusion, by varying the antigens on the surface, you can resolve all relevant kinetic rates of a bispecific binder, both affinities to the single targets, as well as the cooperative binding to both targets at the same time. You can clearly see how both binding arms are influenced by each other, with the cooperative binding being stronger than any one arm alone. What has become clear to the community working with bispecifics is that we cannot predict this avidity binding enhancement or the interplay of two arms in a single construct. Here we condense the rate overview to obtain a so-called hexagon plot, which shows the two affinity and the one avidity binding modes of a bispecific analyte in a very easy way. If we repeat the same experiment with different analytes, bispecific IgGs as well as similar bispecific binders, the result is always different. The effect of cooperative binding depends on the association and dissociation rates of the single binding domains, on the linker length, on steric factors, and on many other parameters. This means that all parameters, affinity and avidity binding, need to be measured every time to allow us to draw a complete picture of the systems that we are working with.
A typical application for an in vitro setup involving the surface immobilization of two distinct targets is in the work with cell surface mimics. In this case, the idea is to translate a typical cell surface expression pattern of two different antigens onto a biochip surface. So this is used, for instance, in the development of therapeutics in order to assay target versus non-target binding. In this case, the target cells demonstrate or exhibit a specific fingerprint of antigen abundance, but also antigen ratio. The most notable antigens, and that we will show you about shortly in a couple of slides, are, for instance, HER2 and EGFR. These are cell surface antigens that are used in order to target various cells in the production of therapeutics. So again, if we wanted to mimic all of these different cell surface expressions, it means that we would want to look at what the relative abundances of these antigens are on the various different cell surfaces and translate that onto our biochip surface. Again, using the approach that I had described a little bit earlier, in the instant where we would mix various different ligand solutions in their corresponding ratios in order to mimic that abundance which is found on target versus non-target cells. And as I had mentioned to you, EGFR and HER2 are two rather commonly worked with cell surface antigens, and for the purposes of these experiments, we'd like to illustrate how you can run these cell surface mimic experiments in order to study target versus non-target selectivity using a standard EGFR to HER2, anti-EGFR, anti-HER2 bispecific antibody construct. In this case, these bispecific antibodies were made with a weaker affinity to EGFR as compared to HER2. And the question here was whether these bispecific antibody constructs had a higher selectivity for cells that were expressing a higher ratio of HER2. EGFR is typically expressed in rather similar ratios for both target and non-target cells. So of course, only targeting that and having a very high affinity to EGFR would mean that you would have a lot of your therapeutic antibody not only on those cells that you would like to target, but also in other tissues where you really don't want your antibody to go. Now, the way that we realize this on the chip surface is we wanted to keep the ratio of EGFR constant in order to mimic that antigen expression. We retained 50% of the ligand mixture of EGFR, so you can see here the bar diagram in order to reflect on the percentage of the ligands on the surface. We fixed a 50% EGFR. In this case, it was coupled to the adapter strand on green, so we wanted to observe again the binding events simultaneously but independently. And then we varied the ratio of HER2 by increasing the abundance of HER2 on the surface by mixing together HER2, the HER2 ligand, as well as those dark adapter strands that I had referred to you earlier that would effectively increase the separation between EGFR and HER2. So this is how we would represent or reflect the relative abundance on the cell surfaces by creating a separation between EGFR and HER2. And of course, at the highest ratio of HER2 to EGFR, this is where you have the largest percentage of HER2. In this case, about 20% of the chip surface is functionalized with HER2. Now, after creating these functional surfaces, we ran the same kinetics assay. So we took our bispecific construct and ran the same assay on all of these different surfaces and wanted to compare what the binding profiles look like. So what the dissociation profiles for the same bispecific antibody looked like. Again, here I repeat, we were looking at the green fluorophore in this case. So we were assaying the enhanced binding when present to EGFR when a higher percentage of HER2 was also functionalized onto the surface. And what you can clearly see when we go from the top dissociation profile that had the lowest percentage of HER2 all the way down to the highest percentage of HER2, you see that there is a flattening of the profile. A flattening of the profile corresponds to an increase in the surface residence time, or the binding half-time that Stephanie had nicely illustrated earlier. Now, what this means is that we are now increasing the percentage of bivalent engagement that we have on the surface. So with the lowest surface densities, where we have 50% EGFR and only about 3% HER2, a very small fraction of those bispecific antibodies are bound bivalently, so the HER2 being the driver really for that strong affinity on the surface. And you can see that over here by a significant surface recovery after a short amount of time for the dissociation. And as you increase the amount of HER2 and you increase that ratio or the amplitude of the bivalent engagement of your antibody, you see that there is an increase in the surface residence time. And again here, which we can see very distinctly by observing a change in the affinity off rates to the avidity off rates by a factor of 100. So just to illustrate those amplitudes in a way that would make it easier as well. So we ran the biphasic fits onto each of these dissociation profiles, again, to see what fraction of the antibody bound was monovalently engaged or bivalently engaged. And you can hopefully see by the diagrams on the right that we have a significant increase in the amplitude of the bivalent engagement. In bar diagrams as well, to put a little bit of numbers on it, we go to only about 40% avidity to over 80% avidity in the case of the highest HER2 expressing surface. Now, what this typically means, we want to go back really to the surface half-times. In this case, one important element or one important consideration in the development of therapeutics is dosing. So dosing and the affinity for target versus non-target cells are both features that are interconnected. In the case where you're only engaging your antibody rather weakly, it means that when you go to very low concentrations or very low doses, you are going to also minimize that non-target cell binding and really only select for the target cells that you're interested in working with. And here we would also like to quantify just to show you what this really means, you know, a change in 100-fold of the dissociation profile. What does that typically mean in terms of a dissociation half-time or a residence half-time on the surface? When we're looking at the non-target cells, or what would correspond to the non-target cells with the lowest ratio of HER2, the half-time for dissociation is approximately 30 minutes, but when you compare that to the surface, which should correspond to the target cell in this case, with the highest expression levels of HER2, we extend that surface residence half-time to over 10 hours. So you can see here that with a stronger engagement, what we do is that we ensure that that bispecific antibody is retained, not only that it is bound to the surface, but that it is held onto the surface, for instance, to initiate any type of downstream processes that are necessary as well in the development of therapeutic antibodies, or for them to actually be functional as therapeutics. So now these assay setups that I had just described to you were really optimized when working with, I would say, moderate to strong binders. You had seen here we had presented nanomolar binders as well as picomolar binders to you, and with standard bispecific constructs that can actually bridge an antigen separation of approximately 30 nanometers.
Now, of course, these assay specifications will align with all bispecific constructs. There are two notable exceptions that I want to show you here. On the one hand, there are weak binders that would require a higher local target concentration to display cooperative binding than we have normally on the adapter chip setup. On the other hand, there are small analytes, such as PROTACs, that just are too small to span the 30 nanometer gap that we normally have between our targets. For these assays, we have established a special Y-structure DNA. This Y-structure is designed to immobilize proteins that are at a lower distance between 0 and 30 nanometers. The individual arms of the Y-structure confine the motion of each of the binding partners to a small volume, effectively increasing the local concentration. One of the design features of this Y-structure DNA is the flexible hinge region, which allows for the unrestricted movement of the arms and can be adapted to different applications. In preparation for this experiment, the proteins of interest are each conjugated to short DNA strands complementary to the separate arms of the structure. In this setup, the reporter fluorophores, the green and the red dyes, are present in proximity to each of the proteins of interest and allow us again to observe the two different analyte binding events to each of the ligands, as well as the cooperative or avidity binding events. One example of a weak bispecific antibody is imicizumab, better known by its brand name Hemlibra, to treat the blood coagulation disorder hemophilia A. In this disease, the essential blood clotting protein, factor VIII, is missing. In healthy persons, it acts as a catalyst in the blood coagulation cascade by bridging the factors 9 and 10 so that factor 10 can react. The formation of a stable blood clot results from the subsequent reactions and is the end of the coagulation cascade. If this factor VIII is missing, the coagulation factors 9 and 10 cannot react anymore and the coagulation cascade stops. Hemlibra is a bispecific antibody that was designed to bridge this gap in the coagulation cascade. It acts as a factor VIII mimetic here. However, once the activation of factor 10 is completed, the blood coagulation factors have to be released from the antibody. For this reason, Hemlibra has very fast association and dissociation rates, resulting in a macromolar affinity constant. The KD of most other antibodies is lower by a factor of 1000 to 1 million, which shows us that Hemlibra is a very weak binder indeed. With the normal adapter chip setup, you would barely see any cooperative binding for this antibody. However, with the help of the Y-structure DNA adapter, you can clearly distinguish between affinity and avidity binding. For this assay, the chip surface is modified with Y-structure DNAs, hybridized with either factor 9 on the red arm, or factor 10 on the green arm, or both at the same time. This special setup increases the local concentration of both ligands to about 150 nanomolar. This corresponds quite exactly to the concentrations found in the blood plasma of healthy adults and allows you to examine this bispecific antibody under physiological conditions. In this experiment, we want to determine affinity rates for both single binding sites as well as the avidity rates for cooperative binding. The Helix instrument runs fully automated workflows from scripted assays. For each new target combination of Y-structure, the chip surface with the anchor strands that Joanna talked about is functionalized with a new Y-structure DNA carrying either one or both of the targets. Then the analyte Hemlibra is flown in and can associate to the targets. Once the association is finished, the analyte flow is displaced by a buffer flow and dissociation occurs. So unbinding antibody is washed away with the buffer flow. After each separate concentration of Hemlibra, the surface is regenerated with new Y-structure DNAs. And from there on, the cycle begins anew. Here are the results of Hemlibra binding to its single targets. On the top, you see the binding to factor 10 alone in red, and on the bottom, you see the binding to factor 9 alone in green. You see on the first glance that the dissociation from factor 9 alone is much faster, with only a few seconds overall residence time, which leads to a micromolar KD. The binding to factor 10 alone is a bit stronger. Here the dissociation takes about 2 to 3 minutes, and we get a KD in the middle nanomolar range. If we now present both targets to Hemlibra at the same time, the binding changes a bit. Here on the bottom panel, you see the binding to factor 9 in the presence of factor 10, and on top you see the binding to factor 10 in presence of factor 9. Most notably, the presence of factor 10 changes the binding to factor 9. Those are the diagrams that Joanna showed you in the beginning. And as she already told you, here you can see the very strong cooperative binding that changes the dissociation profile from factor 9 by a lot. In contrast, the binding to factor 10 doesn't change notably in the presence of factor 9. If you look at the rate overview, the explanation becomes quite obvious. Here you can see again on the left the binding to factor 9 alone, and the fast part of the binding to factor 9 in presence of factor 10. The binding rates correspond nearly exactly. In contrast, the slow dissociation of factor 9 has nearly the same dissociation rate as that of factor 10 alone. This shows us that the contribution of the very, very weak binding partner factor 9 doesn't change the binding to factor 10 at all. So in conclusion, now these rates that we could resolve here from this very weak binder could not have been measured with the normal adapted chip setup, but only with our special Y structure.
Right. And with that, we hope to have highlighted for you the most prominent bispecific applications made possible with the Helix biosensor. The DNA-based surface functionalization awards the flexibility needed to adjust the surface density to enable you to assay targets of varying size and also to immobilize different targets in variable ratios, maintaining the same assay orientation to measure affinity and avidity binding of bivalent analytes. And of course, a key component to complete the assay is the analysis of the readout to extract the corresponding binding rates and kinetic parameters. This is, of course, made possible thanks to the multiple device features that automate the process from generating your desired surfaces in the various configurations of target ratio and density to the powerful data interpretation software Helios. The microfluidics system enables you to work with very high flow rates to assay weak binders without rebinding effects, and the sensor surface is stable enough to allow you to run dissociations for many hours. This expands the range of binders that you can assay from the sub-picomolar range to the millimolar range, either using our standard DNA setup using the adapter chip or the Y structure solution that Stephanie just presented to you. And of course, last but certainly not least, our dual color capabilities for the simultaneous observation of multiple surface binding events.
Thank you, Stephanie and Joanna, for the detailed presentation. In the time remaining, we'll address questions from attendees, so please continue to add questions to the chat window, and we'll address as many as we can. I have a quick question first. How much assay development time is required for each of these binding experiments?
In the assay development that we did here at Dynamic Biosensors, we focused on an ideal reproducibility of the workflow for different analyte systems. So what you need to do generally, or what what we do generally if we get a new analyte, is that we need to conjugate the targets to our DNA adapter strands or to our DNA ligand strands. This is a very standardized workflow that works quite well in nearly all cases and takes about half a day. Once those target strands are ready, you can already start your experiments. With our very standardized workflows, you don't need to put much time into assay development. In some cases, you need to adjust the surface density to obtain ideal biphasic curves, but mostly our standard workflows work quite well. So what you need to do is to test a concentration series. This is quite a short screening process because you only need to observe the association, and then you are already ready to go. Then you need to measure a suitable concentration series. The dissociation time depends a lot on the antibody you need or you want. And that's already all. So normally, I would say that depending on the dissociation time, it takes about 2 days to 1 week to measure a new analyte and to determine all the relevant rates.
Thank you. What is the lowest density you have to choose for so if you're using a small molecule, for example, how low a surface density can you go with, and how do you know what that surface density is approximately on the chip?
So we measure surface density, or we monitor the presence of ligands on the surface using the fluorescence signal. Now our photon counters are highly sensitive, which means that we can work with very low ligand densities, achieving target separations of approximately 300 nanometers on the surface. So we can go really to very low densities. Here we don't necessarily express it in terms of a concentration because we're already working on a surface, so it isn't a quantification in that sense. We talk about the individual ligand separations or the interligand separation that you can achieve, and this is down to about 300 nanometers.
And as a follow-up, how do all these things change? How do these considerations change if you're mobilizing something larger than a small molecule? So the small molecule in the question was on the order of 460 Daltons, but what if it were an antigen of 20 kilodaltons?
So yeah, thanks for that question. So the typical antigens that we are working with, and as we had mentioned for all of these assays, are larger proteins on the order of 60 to 80 kilodaltons, and this is typically the standard size that we are working with. Now our automated workflows are created in such a way that you can select a standard ratio. So this is a recommended ratio that you get of your ligand to the adapter strand. It is flowed over the surface, and then you can see immediately from the profile that we have on the surface how the kinetic profile or how the dissociation profile actually changes. And from there, that will give you information as to the amplitudes or the relative ratios of the monovalent to the bivalent engagement. And from there, you will be informed as to whether or not you need to increase the density that you are working with or effectively decrease it by adding adapter strands into the solution mix.
Thank you. Here's one that's near and dear to my heart. How does this technology work with DNA binding proteins?
Thank you for this question. So this is really an answer that is very dear to us. Of course, you can also work with DNA binding proteins. As we are having a DNA-based platform, of course you can conjugate different DNA strands to our working DNA strands that we use. So DNA binding proteins are really no problem for us at all. It is very easy to observe these proteins, and our surface is made for them.
Thank you. So could target or oligo conjugation block the binding site, and how would we deal with that?
So yeah, that's, typically a question that we do get as well is there a possibility that the actual conjugation itself may impact the affinity of the antigen itself to the antibody? One way that we, or one approach that we recommend using for all scientists or all users of our device, especially when it's the first time that they run their assay, is to invert the assay orientation. So first to work with the ligand on the surface and then to work, for instance, with the antibody on the surface and see if the kinetic or the binding affinity is comparable. If it is, then that is an empirical indication that we haven't necessarily blocked the binding site by running the conjugation, but then if there is a significant disparity, then of course further analysis then is needed in order to confirm whether or not this is the case. And alternatively, if capture approaches can also be used on our surface. I know that I didn't go into that into much detail today, but another approach would be to capture your ligand of interest or your target antigen of interest rather than conjugate it. And again, here then using the same assay setup, you can also compare the binding kinetics, that which is conjugated and that which is simply captured using a his tag, for instance, or Biotensis strict avidin capture. So those are the options.
Thanks. So there's a follow-up to the DNA binding protein question. They meant really we're interested in looking at protein-protein interactions for proteins that also bind DNA. So how does the technology work in that situation?
For situations such as this, where we have some kind of unspecific binding to the DNA strands that we use to fix our proteins on the surface, for this purpose we use reference curves. You can either use a reference curve as a blank injection, that you just do your experiment again but injecting buffer instead of your protein, or even more elegantly with our helix system, you can use the second electrode as a reference electrode. Like this, you get a real-time reference from protein binding only to all the DNA strands that were used in the experiment without the protein that is your original interaction partner. And those reference curves can then be used to correct your signal for any unspecific binding. And like this, we can control quite reliably for unspecific binding to the DNA strands that we use.
And let's see, can you elaborate on antibodies binding at the same time to both targets versus antibodies that are tumbling along the targets?
So by tumbling along the targets, is this a binding dissociation, rebinding effect?
So the question says, of course, slow down it's something that slowed down dissociation. Perhaps the questioner can elaborate on that a little bit, and we'll get back to it. There was an earlier question, how flexible is the platform to onboarding new analytes? I'm assuming that means different types of proteins, things that are not proteins. So I guess the diversity of analytes that we can target might be the question.
Mm-hmm. In terms of the analytes, we can actually assay a very wide range of them. So we can, work with both both assay orientations. The analytes themselves can be anything from small molecules to large bispecific constructs, as we had indicated over here. It's not a mass-based readout, it's a fluorescence-based readout, which means that it is not mass-dependent. And so, and as Stephanie had already mentioned about, we have an in-channel inflow reference electrode, so it would be, I would say, rather easy in order to onboard all of these different analytes, because you can immobilize your target, for instance, one of the binding partners on the measurement electrode. The reference electrode does not carry the binding partner, and then you can assay all of these different analytes that you have to see what types of signals you're getting for the specific versus any potential unspecific binding events. But again, because it's fluorescence-based, we are rather independent of the mass of the analyte that we would need to work with.
Thank you. So the previous questioner has clarified the by tumbling, he was referring to including rebinding events. So I guess it's looking at antibodies binding at the same time to both targets versus rebinding events. And how do we differentiate these different types of binding?
Thank you very much for the clarification. So for the rebinding events, I have to say normally we really try to avoid them. Our helix instruments allow a very fast flow rate. This enables us to avoid rebinding altogether. Normally you see rebinding as, let's call it, impurities in your curve. So the curves don't correspond exactly to your model anymore. So either the first part of the curve is not in accord with the fitting curve that you overlay it, or the second part, for example. And if you see such effects, you really shouldn't fit your curves, but remeasure them at higher flow rates. So for example, if you see linear parts in your curve, this means that the 2-to-1 model that we applied here doesn't correspond anymore, and it would be incorrect to extract any kinetic rates from those curves. So yeah, to the rebinding events, we really try to avoid them.
Well, thank you. I notice it's coming up on the hour now, so that's about all the time we have today. If you squeeze in another couple of questions, we'll certainly get back to you by email. I would like to thank our speakers, Joanna Deek and Stephanie Mack, Cambridge Health Tech Institute for hosting this webinar, and to all of you for your attention. If you have additional questions or if you'd like to have a more detailed discussion, please contact us at the email addresses listed on the final slide, and have a great rest of your day. Thank you, everybody.